Hi, everyone. My name is Amir Amedi. I'm a lab as part of ELF ICNC. My lab is located in the medical school and also part of cognition. And we spend, I understood, the next, check it out, the next 2 hours together. And I hope we'll have fun. So you are in the second year. I understand you just finished several lessons about vision, right? And can you tell me in 2-3 sentences what you were? Did you cover retinopathy, for example? Retinotopic mapping? Did you talk about ways to predict activity from the brain based on the representation in the visual system using support vector machine or things like this? You still okay? Got it. But roughly speaking, okay. So I'll tell you what I will try to cover today. Today I'm going to try. I think, like in the previous lessons about vision, you learned the textbook kind of and like advanced textbook way that most of the people look on the visual system. What I want to do with you today is share with you a line of research which tries to argue that the way we think about visual processing in the brain and visual organization of the brain, while it's the most studied topic, basically vision in neuroscience probably so far, there might be some alternative views to how, I'll try to share with you an alternative view of how the visual system is organized and what's the driving power that drives the visual system. So this would be one line of work and of research that I will try to share with you and we can have a little discussion here, or you can fight and argue, and so it's still all relatively new, and everything I'm going to show you, like over 90% is already published, but like always in science, there is always, sometimes it's very difficult to bring together different lines of research and different types of results, and this is one thing that we will try to do together. And actually, I will try to generalize and to suggest that what I try to show you here is true for all the sensory system, or at least all the topographic systems, which are mainly audition, somatosensory system, vision, and the motor system. Okay, so this is one thing that I want to share with you today. Can you see the screen? It looks like very bright. What? How can we? Better. Cool. Do we have here a sticker? No. Okay, it's a small class, so we'll do it. Yeah. Okay, so the concept of topography, and actually this is something that is true both in animal research and electrophysiology, but MRI brought it to a whole new level of verification. And the notion of topography is the concept that in the brain, from the level of the receptors all the way to the levels of maps in the brain, neighboring characteristics of the physical world are represented in close topographical ways. So in a way, you have a given topography that keeps, with some modification and some distortions that are supposed to serve us. We can. No. So in the auditory system, for example, it's very simple. The basic representation in the cochlea is the frequency and the pitches. So T. Okay. So the topography in the sense of the maps in the brain means that I'm going to have a very organized map in which the high frequency will be here, and then the lower and lower and lower. Actually, I have a whole. Wasn't what I planned to do with you today, but there is a whole presentation about how much actually how much the brain is topographically organized. Okay, and there is not a lot of computational work that show what is the computational advantage in having the maps and the neurons organized in this way and represented this way. But it seems that something in the short and medium range connection, there is some optimality in the way that there is topography. I might show you a little bit from a work we did on the sensory and the motor system. So for example, there in the body, the concept of topography also, it's very clear, right? Because we have all the topography in the body, and then we have the homunculus, okay? And using Fourier analysis as phase encoding, in which you encode neighboring body movements in an organized fashion, in the scanner you can show actually that there is over 12 homunculi distributed across the human cortex and subcortical regions. Actually kind of fun, so maybe since it was raised earlier on, I can show you a couple of them. And actually you can also do, and we can talk also a little bit about that, you can also train the brain to be kind of a decoder, okay? So basically it's all very fancy words to something very simple. Let's say I have two voxels in the brain, 1 and 2, okay? And let's say one is responsive to my feet or to a low tone, and one is responding to my mouth or to high tone, okay? So basically I can show a lot of movements to this, I can call these two voxels my classifier, and each time, let's say I'm now moving my feet, and there is a lot of variance, of course, noise in the brain and so on and so forth, but now I'm moving my feet, this area is going to, voxel is going to respond high and this low, right? Next time I'm going to do it, maybe they will be like this because there is some noise, then like this, then like this, then like this. I'm going to repeat it many, many times. Then I'm going to present the face and I'm going to have something like this, okay? A lot of sampling. And now I decide what is the optimal line that separates the ratio between these two voxels. So now that I get the data from the brain, I can predict whether the person moved the head or moved the face, okay? Or whether the person perceived a low tone or a high tone, right? Now, in two voxels and 2 dimensions, it's very easy to think about it. Because basically all you are doing, you're having basically voxel number 1 and voxel number 2, and you get some distribution of the activities, okay? And then, let's say this is all the areas that the voxel 1 responded high, let's say for the high tone or for the face, and this is for the low tone or the foot, okay? So I'm going to try to have a line that separates the best between the two situations, two states, high tone, low tone, or moving the feet or moving the face. And now I'm going to have a new data point coming from the brain. I don't know what the person is doing, or I'm recording it, but I want only to predict from brain activity. Okay, let's say I'm getting something like this. Okay? So now the decoder line tells me there is higher chance it's arrived from a high tone or from the face and not from the foot or low tone. Of course, if I'm getting a dot here, then I can really say, you know, there is very good chances. And all this decoding method says is that, okay, now the situation usually is, okay, this is an easy example. So we, for example, what we try to do is to have 20 different body parts. Some of them are very similar. For example, can we decode a person playing the piano while he moves fingers? Okay, can we predict which finger he moved? And that's what we did in this study. Okay, so we have the maps of the homunculi in the brain. I might talk a little bit about that as an intro to something else, but I will explain a little bit because I think it's a very important concept. And now there is some researchers, very few, but some researchers, and I think it's become more and more common with them than it used to be, is that maybe also higher level concepts are also represented in the brain topographically. Okay? Just before I'm saying this, So now if I want to do between three areas, between three voxels, and have the prediction between let's say three different fingers, then now I have basically a three-dimension, and I need to have a plane that separates between the three states, okay? The level in each of the three voxels. And the generalization for more is just, this is why you use support resolution. Basically, you try to reduce it you get a mathematical solution that gives you the best prediction according to your training data, okay? So this is the concept. Now, let's say that we have representation also for high-level functions. So for example, let's imagine, so first of all, we know that in visual system you learned there is representation of space. So when I focus now on you, all the right side, dot by dot by dot, is representing my left side hemisphere. Okay, and they are neighboring. So first of all, I'm going to have in the most posterior part you, and then I'm going to have you, right? And then I'm going to have three empty chairs, and then I'm going to have the wall. All of it represented this way. And all the other side this way. And not only this, we don't have only one map, we have multiplications of these topographical maps. Okay? Exactly. This is actually what defines the visual areas. When I want to now study V2 or V1 or V3 and understand for their characteristic, what I will do is I'm going to do a topographic mapping, and I'm going to find, let's say, I think it's a Chazara on the previous lesson, but it's so beautiful that it's the same in all the systems, and that's what starts to emerge the picture. So let's say V1 represents the fovea, yeah, and all the let's not get into all the details, but there is also another representation of the vertical and horizontal meridians. Okay, so one is going from the fovea to the periphery, but also if we take the visual scene and represent it to 360 degrees, then first we start with the 90 degrees on the horizon. Yes, so first if I'm focusing on you again, then I'm going to have all this line represented in the calcarine sulcus, all the things that are above by degrees going in the lower part of the V1, and all the places that are below on the upper part of V1. So I have a topographic map of the world. It's distorted because, for example, in the body system, we have, actually there is slides on all the things that I'm saying now, but basically it's distorted in the way that things that are important for us for a given task or function are going to get a lot of real estate, of neuronal real estate. And things that are less important for us are going to have less real estate. So for example, there is more neurons dedicating in this body map for the thumb than the entire space of the belly and the back combined together. Okay, so in this way the map is distorted. Okay? Or there is third of the visual system, because it's all come together, because the just noticeable difference, the resolution is the best in these places, and it's high because we want a lot of neurons to control them and to have a very high resolution, because it's important for us for a given task or function. Okay? And the way we actually study this, if we want to study a given brain area, then we are doing a mapping of all the 90 degrees, yeah, up and down, and we have all the representation here. And okay, and now let's say it's something like this: the horizon, and then go up. The lower part of the visual field will be higher in the calcarine sulcus. And then I'm finishing to represent the entire space, okay? Now I have a mirror symmetry map that starts from up and go down. And then another mirror symmetry map and another. And this is actually how we actually separate the different visual areas. The beauty of it is that also in the auditory system, when we are looking at high notes, it's actually more easy to understand it in the auditory or in the body systems. Yeah, so for example, in the auditory system, if we are going to have high tone, okay, and then there is all the topography, and here you reach the lowest tone that we can recognize, then the next map is going to be a mirror image of it in both sides. Okay? So if it here went from high to low, then here it will go from low to high, and this is how I will define the borders between the areas. Okay? Or in the body, if I have the map going in PCG, in M1, let's say, okay, and SMA. So if I have in the midline, what representation do I have? Which foot, exactly. So if I have the foot here, and all the body goes laterally, right? So I have another map that goes from the foot to the face again, which is a mirror symmetry. Starts from the foot and goes to the face. And this is how I define the border between M1 and SMA. And if you would use other advanced methods, like, for example, today you can map the population receptive field, the average receptive field of a given neuron, you can actually see that both in the visual system, in the auditory system, and in the motor system, what happens is that we have multiplication of the map, but the receptive field expands. Okay? So this is one line of a... Actually, I do want to expand a little bit on it, on one more concept. And this is the concept that, okay, I showed you so far, and this is actually is agreed by, I think, most of the neuroscientists today, that the basic organization of all the senses and of the motor system is very topographic, one. Two, that there is multiple representations. Usually, when they are very neighboring, it will be a mirror symmetry representation. It helps us to define the areas. And there is multiple such maps for each system. That actually spans most of each of the cortices, auditory, visual, somatosensory, motor. Now, some researchers, and this starts to be a bit more speculative. First, the notion that there is many maps for each modality starts to emerge in the last 2-3 years. It was not sure if there is 2 or 3, but it seems there is over 10 for each of the modalities. Okay, so we'll mention this, but there is even more important concept that I think also is coming. Its type of research is more easy to do in humans than in other models. And this is the notion that maybe also the higher functions are organized topographically. Okay? And I will give you 2 examples. Okay? One example, let's take this example of predictions and our ability to predict from the activity from the topographic maps. Okay? Now, or let's go to something actually more simple, and this was something that was shown already, the concept of numerosity. If I'm going to project to you very quickly a lot of dots, you have a sense for quantity, for numerosity, even if you don't count each number, right? So up to some number you can do it very accurately. Up to 7 you can do it without counting very accurately, but also beyond that you have an ability to estimate without counting. Okay, and there is a researcher, Domlin, that showed that this concept of quantity or for numerosity is represented somewhere in a topographic map in the parietal cortex. Okay, and he can predict what the person was seeing based on the activity of this map. Okay, now let's start to speculate a little bit. Okay. And I think this is a very, very part of my goal for these 2 hours. It really doesn't matter if we do 5 more slides or less. I want to open to you to various fields of research that I think are in a way exciting. And when you think about your field, doesn't matter in which method, you will be open up to this several other new concepts. Okay, that was at least my goal when I. Okay, so now let's say we have phonemes. Okay, right? When we talk or when we read, we have phonemes. Let's say, I don't know what, tell us about the future. Okay, so we have phonemes. Now imagine that there is a topographic map in the brain in which all the phonemes are organized, one next to the other. For example, in semantic, we know many times that when I want to retrieve a given a word from memory, many times I will do a mistake and I retrieve a neighboring word in terms of the concept, right? Yes. Do you see what I mean? Okay. So if I would want to say football, sometimes I would say basketball, okay? And the theory behind it is that these representations are close to each other in the brain, okay? And then there is some, it's more easy to do this mistake than to say apple when I'm thinking about, okay?
This is true, it says something about the topography of semantics and not of the...
Definitely. I just wanted to give you, to say this is not such a far-fetched concept, idea, okay? Now let's assume, now we are doing a thought experiment, okay? Let's assume now that there is topography for the phonemes, okay? In the brain, okay? Now, let's say I'm mapping this, and now I try to predict when a person would say pa, or ga, or da, based on the activity on all this topographic map, right? So this is doable in principle, right? Great. Now let's say this is doable. Now let's say I'm saying to myself, while I'm in the scanner, I'm thinking some thought. I'm saying to myself something. I need to go tomorrow to the dentist. Okay? Or I really like this person. Now, in principle, we can decode it using these decoders. No, I'm telling you what was done. You asked me about topography, okay? And I have a couple of slides there. It wasn't the main topic, but because I wanted to talk about plasticity, but I think it's a very interesting topic, and I try to connect it to what you learned in the previous lesson. But if it's true, and so far we have probably only this example of numerosity, of high level, really very clear topographic map. But you need to understand that, for example, the multiple homunculi in the motor system was done this last year by a group. Okay, not Charia, by the way, ICSC student that worked on the lab did this work, and the ability to predict the motor movement based on the MRI activity. Okay, and if it's true, also you can do the same with if you have electrodes and you think that there's something that is higher level than just vision and the height of the tone. Let's say in the auditory system you think about something more complex. If it's organized topographically, you can also put electrode and retrieve the mind or read the mind of this animal. Okay? It's not limited to imaging. Okay? Yeah?
Why is this related to topography? If it's randomly distributed,
If it's randomly distributed, you have a problem. Why? It will be hard, right. So in principle, all the people that have neural records usually till now, and now there is a start of serious work that actually was driven a lot by Google's great ability for showing learning in neural networks now with deep learning, etc. And it seems there that the topography plays a role. So, but usually most of the neural network guys that I was speaking with them about this idea a couple of years ago would say what you said. It's nice that it's organized topographically, but in principle, it can all be just random. It doesn't have, it's the same. I can shuffle all the connection and all the, but I think there is something deep computational that you guys need to solve, okay? Because the brain behaves like this. You cannot argue. Most of the time, all these senses are organized this way, the motor system organized this way, and now there will start to be a question of how much the higher level functions, emotions, other stuff, are also encoded topographically. If yes, then yeah, your life becomes much more easy. Especially when we talk about methods that have much poorer resolution than electrophysiology. Because now we want the whole column to respond to in the same way, and if they are organized next to each other, then actually even you don't need the exact column, but you need the relative weights across the 20 or 1,000 or 10,000 voxels that you have. You just need the relative weights between them to predict what the person is thinking or planning or doing. Okay? Okay, so this is one topic I want to share with you. The second topic is this alternative view of how the visual system is organized, and what I will try to argue that, this alternative view of how the sensory systems are organized, and again I will focus on vision, come from actually a work that we did on brain plasticity, okay? And we characterize two types of plasticity that usually can happen in the brain, in the large-scale organization of the brain. So the two types of plasticity that I'm going to talk about is the following, in the level of the maps. Okay, again, I'm talking about the large-scale organization, the maps, the topography, the specialization for given functions, like you learned, I'm sure, about selectivity for body, visually selectivity for faces, selectivity for reading, and so on and so forth, right? So beyond the topographic map, we have further specializations to given functions that are really important for us as animals. And then you might have two types of plasticity. One plasticity we can call task switching cross-model plasticity. Okay, why do I talk about this type of plasticity? This type of plasticity says a given area has some function. It responds to high tones or low tones. It responds to faces, or it responds to written text or to phoneme, whatever. And now something happened in the brain or in the experience or in the learning process of the person, and now this area is doing something completely different. Okay? One example that I can give to you here is a study that we did on memory and the memory capacity of individuals. And we found out that people that are early blind, but not late blind, can have their topographic visual map that are basically don't get the information from the eye, right? So they are unemployed and they don't get the input. Then basically these maps can actually now start responding and be an integral part of the memory system, including the hippocampus, parahippocampal, and the parietal cortex, work together with them. And even more than this, there is variability among blinds that some can recruit very little of the visual system and some recruit almost all the maps. And there is a very strong correlation between how much they recruited to memory and how much superior ability they have. And even more than this, you can interrupt the activity there and interrupt the memory task only in blind but not in sighted. So in this case, you have a situation in which a brain area was responding to one thing, to vision, and now it's responding to a completely different task.
How do you do that?
That's a good question. How did the brain do it? We don't do anything. We just...
some sort of specialized training or?
No. This is something that we didn't do any... I would show you some training stuff we did. And actually, the other type of plasticity is something that's very easy to do with training. This was just inviting them. We actually didn't track the process of it because it started presumably... we did one study, which we are writing up only now, in which we took sighted people and we made them blind for 5 days. Not really blind, we just put a blindfold and a little film inside, and it was done together with in Harvard Medical School, because there they have a whole wing that you can put patients that are not really patients, that are for research, okay? And you have the nurses treating them and, type of resources we don't have here. So this was a collaboration with Alvaro Pascal-Leone. And then we basically replicated the study that we did on the memory and several other things. We taught them to read Braille, the sighted people, and several other things. And basically we showed that even after 5 days, you start to get very the beginning of such processes in some areas in some parts of the map, more in the periphery, less in the fovea, but you start getting, and I will show you some data from our lab about navigation that shows the same thing. So how this is happening, we're only now starting to have answers, and I would try to argue how this can be done, more for the second type of plasticity, but this is a very good question, and we need to treat it. Okay, 2 quick questions, and then I'm only in the second slide. Yeah?
Losing functionality. When you say cross-modal plasticity, you mean that you lose the functionality you first had?
Right, so that's a good question. So where does this question become very, very important in medicine? Can you try to think about a situation, a medical situation that you will really want to have this type of plasticity?
When there is damage to
Exactly. For example, what's the most common thing that creates damage to a given brain area? Stroke. Exactly. So there you have an area that had a function. You lose the function, and now you need to convince a different brain area to take the function. Now, also maybe you raise this question. If no, I will raise it. The question is, one, can we convince, and how can we convince? This is still an open question, because in our case, they had new experiences during the critical period. So maybe we know it's more easy for the brain to change tasks and to change functions. The blindfold study suggests maybe some capacity is also there, even in the old brain. Okay? And some of the stuff, if I have time, we'll see, that we are doing right now. But second question is, one is, can we convince a different brain area to take the task that was lost? It's already actually strokes show how much there is division of labor to different tasks, because otherwise you have a general reduced. There was some theory, for example, actually, some that say that all the functions are distributed in all the brain, and you would just lose a little bit some of the function across the board. But strokes showing us this is not true. Okay? Now, the second question, if we are going to convince a different brain area to take the function, does it mean that we are hurting the function that he is doing before we are convincing him to take the function? We have some data to suggest that maybe yes. It might be an issue that we need to think about. Okay?
So have a dual functionality, like little less than this, little less than this, but at least function.
Yeah. So it's possible, but it's very tricky. And this is, I think, something that is not understood enough. And this is, I think, another field. I want to open for you fields which I think are exciting in the next couple of years, to my humble opinion. Each one will do whatever he wants to do. Okay. Second type of plasticity. The second type of plasticity is similar in a way, but it's in some ways it's very similar, in some ways it's very different, and it seems like the mechanism is completely different, because it seems like that this type of plasticity can happen in no time. Can happen very quickly and even in the adult brain. And this is what I will try to show you here. And this is why we think actually the whole view about how the visual system or the sensory system are organized is not true, or it's different, or we can argue about it. Okay? So this type of plasticity says that if a given area has some function, for example, let's say a given area supports reading, supports phonology, supports face processing, supports tool manipulation in vision, or in one sense, then there can be a cross-modal plasticity which is switching the inputs that is driving the function, but the task is maintained. Okay? So we call it task-specific sensory-independent cross-modal plasticity. So the concept here is that, let's say I have an area that I use to read this word, task, or recognize this face, and we know that there is specialized area there. Then, if I can convince the brain to read using a different sensory modality, for example, can you give me an example in which I can read the word task using a different sensory system? Braille, exactly. So the question is whether when I learn to read Braille, I'm using the same real estate that used vision to read, or I would need a different real estate to create a different new representation. And what we show is actually information goes from S1 directly to this visual so-called area and take this over. But I will try to show you this is generalized to any functions that we tested so far, or you can think of in vision. Well, we didn't test any, but almost any that is known, and it seems that this is the case. So for example, when you navigate now, and you recruit some of your visual system, the dorsal stream, to navigate, now if you start to navigate using a different sense, the brain can switch the senses very, very quickly and maintain the function. And this is true even in the sighted brain, even in people that didn't have any deprivation. They just learned to do it using a different modality. Or the example, what will happen now if I teach you to read Braille? Okay? Okay. So we also try to start to see how can we look into question of mechanism, connectivity, etc., even when we talk about the human brain and studying humans. And which type of visual deprivation derives the two types of plasticity, and so on and so forth. Okay. Now I like also to give a couple of take-home message, and then we really can start. I don't know what's the time, but we will start in the second lesson. Okay. But for me, this was already the bottom line, right? If you got this, now I just need to convince you, okay? Or we can play with some data. But the bottom line is that we see with our brain, not only with our eyes. This I hope you already got this in the previous lessons on vision, but if not, I would try to give you a couple of more examples. Using this, we can actually teach the blind to see with the ears or with the hands with almost any visual concept. We will test visual brain areas, including center reading, object face, depth, navigation. And actually, I want to show you that all the centers, we call them visual, but actually they are if you teach the brain to do any of these tasks using a different sensory modality, very quickly the response profile would shift into this sensory modality. Okay? So this is one type. So this goes to this new theory that the brain is not a sensory machine. It's not divided by the senses and by this specialization, but by this task. What is important is the task. The task is mapped usually to some certain topography, and the brain can switch very quickly between the senses. We can use this also to develop new technologies. Another take-home message is that if this is true, and any input can drive any brain area, even after passing the critical period, then it means that actually if we put multiple information, especially when the situation of the signal to noise is low, for example, when I try to see in a dim light, or I try to read to understand what you're saying in a different language, like now I'm speaking English, not that great. Okay, so for example, when I moved to do my postdoc, I had no problem to interact with people in English, but when I had to sort out something in English on the phone, the situation collapsed. Okay? Why? This is because of the McGurk effect. Okay? The McGurk effect means that we integrate both the movement of the lips and the sound to understand the phoneme. Usually we think about this as an auditory ability, right? To understand a different person. But no, it's as much vision as auditory, so there is benefit to combine information. So maybe for rehab or for driving, to convince a given area to take a function, then we need to put information, overlapping information from different sensory modalities, then we can make the rehab much better. So this is another bottom line. And the last type of, I would skip this. This is, I think, there is no chance we are going to get to this. The next thing the last thing that I would try to tackle in this hour is this notion of critical period and how much it is. I think experiments by Hubel and Wheeler were very convincing, but the thing is that they were performed on a very, very specific type of input and situation, which is very, very low level. They look on the hand high dominance, and so on and so forth. So the question is, how much do we have a hierarchy of critical periods, and how much with enough effort we can actually convince the brain to change even after the critical period. And actually I would show you that even with zero visual experience, we can develop all the topography and the selectivities that are part of the visual system, all the high order, and we have some data to suggest that even the topographic retinotopic areas are as flexible even in the age of 40 or 50 or 60. Okay? Okay, so let's start. Until the break, I'm going just to show you a couple of fun examples to the notion that it is not just taking external world, right? When we want to see, we think about, when we talk about vision, we talk about, we have electromagnetic waves that are between 400 nanometer and 700, 800 nanometer, and this is spectrum give us, we can see the spectrum, right? Because we have receptors here that are topographically organized. The receptors maintain a reflection of the topography in the outer world, and if it falls in this spectrum, then we are going to have neurons firing and delivering this information to the brain, and this is how we see, right? So it's a very mechanistic way of thinking about vision, okay? So we have an input, we replicate the input, and we travel the input to the brain, okay? Now let's look at this little example that I'm sure you all, where your friends bother you about this. And as a brain scientist, you have to tell them, I understand this. So do you know what happened in the... First, let's do a little experiment, okay? Because it's actually very different in different lighting conditions. Let's see first of all if it's working here. So let's focus on the middle dress, okay? And I, for example, see this dress to be white and gold, okay? Now, anyone here see it in a different color? Anyone see it in blue and black? Yeah. Blue and black, blue and black, blue and black, blue and black. The middle one, yes, the middle one. Okay, so 5. See, so we are about like 2/3, third, or half, something like this, okay? Now, what's happening here, okay? We said that the light is blue supposed to be 400 and gold supposed to be like 600 nanometers. So how can it be that half of you, and when it started to be on the internet and celebs started fighting over it, then they say maybe there is different images that run in the internet. And actually when we look for it, it's true. There is some images were like this, and we will go back to this, some like this. But this is not a story. It was not a fraud, okay? It was a real thing, because here we did this in the same lighting condition, in the same input. Some see one color, blue, and the other see a different color. So this is already ruined everything we said so far, that what we see is the wavelength, right? So let's try to explain what happened here. And now you can go. So actually we understand half, and half we don't understand. So this is almost always the situation in science. You really understand only half. You can pretend that you understand more, but usually you understand half, and there is half new questions opening up. So let's see what happened here. Let's start with something that all of you see the same, right? If we take this line, I guess all of you see this part of the line to be darker, and this is lighter, right? And you know it's the same color, and actually I take it to the side, and you know that it's the same color. And who it doesn't, he needs to go to the eye doctor, right? Okay. Now what happened here? It's very simple. If the background is bright, then it will make our brain like to see contrast, and the neuron respond to contrast, so it will look this part darker. And if the background is dark, then we are going to see this line as brighter. How does it help us to solve the story of the dress? Well, actually, this is what happened with the dress. And actually, let's see another situation. This, I think, also you know very, very much. A and B, actually, this is in white, and this is the letter B in black, because you have impression that this is black and this is white, but actually, it's exactly the same grayscale level. Okay? Exactly the same. So the environment also triggers, the context triggers the grayscale level. Now let's explain it with the colors. Now what happened? If the ones that saw the dress is black and blue had the illusion that the dress is in the shadow and the environment is in very bright light. So then your brain, your visual system, which will soon show that it's not visual, but that will wait for the second lesson. Your visual system remove the blue cast and the darker part of the image, and you perceive the image as white and gold. Okay, so if you think the dress is in the shadow, the brain will remove the blue and the black, the darker part, and you would see it in the brighter colors, and this will look as white and gold like me.
But why do we have differences? Because we
So I said we understand half and half we don't. So let's first finish with what we understand. Now, if I think, those of you who think that the dress is washed by very bright light, and the background is more dark, then what the brain will do is it remove the whitish, the bright colors, the white and the gold, and you would see it as a black and blue. Okay? So now we have half of the answer, and what is the other half that we don't understand? Why, third of you, their life experience make them think that this dress is washed by very bright colors, and the other half have an impression that this is a dress that is in the dark. And probably the answer is that all the life experiences that you had so far made you translate or interpret the image in different ways.
But we
Yes. So just to summarize, and there is a lot of other examples that I really like to give is this example of the FedEx. I saw it many times, submitting papers, and then one day I noticed there is an arrow in the FedEx. So how many of you knew this logo, but now see for the first time that between the E and the X there is an arrow? Raise your hand. Okay, so more than half of the class. So, and you knew this logo many, many times. Now, it again showed the amazing plasticity also of the system. Why? Because one trial I did. Now, next time you go in Paris or in Tel Aviv or in Jerusalem and see a FedEx truck, the arrow will jump on you. You cannot get rid of it. No. Actually, I looked after that and I found out that I wondered if it's a shitty logo and that nobody that listened to a boring lecture and the guy that designed it did it in purpose and actually won some design competition for it. But at some audiences, this is a sophisticated audience. In some audiences, I'm giving the talk. I would say the average is that over 90% of the people knew this logo and didn't see the arrow. So to summarize this point is that we see with our brains, I won't have time, but I have a lot of examples also from the other senses to show you it's the same, is that we also hear with the brains, we also touch with the brain, not less than we see with the eyes, and maybe we can use it, okay? We can use it for research, and we can use it for rehab. Okay, so let's take 10 minutes break, okay? Because I have a lot of stuff still to share with you, and we had a lot of discussions. We have one last discussion. This is on your break, yeah.
The article is only on the middle dress because it's both on the.
We actually can, in some cases, there are people that are. Let's try to see, okay? The left dress, anyone can see the. Let's go to the right dress. This is a chance that it will work. Anyone here see also the right dress as white and gold?
Half and half.
Oh, almost, yeah. So it's interesting. Sometimes there is people, even and sometimes there are people that see this dress as blue and black. And what's the difference between the two? The brightness level. So here you increase the brightness level a lot, and now the illusion is that it sees that really both the background and the dress is in very bright colors, okay? And so in here you can see that the contrast, there is a bigger contrast. So if you play with these things, Actually, if you go with Photoshop and check what is the hue level here, the average, it's actually brown. So actually the real color, the real spectral color, has nothing to do, not with blue, or not with black, not with white, and not with gold. Okay? Okay, so with this philosophical thought, we'll take a break and we meet here at 5. Okay? 4. And we have until 5, right? Okay. כן.
אז אנשים עיוורים מלידה שיכולים להיות טוב באופן כללי מאנשים...
כן.
כן?
כן. גם שעשינו גם את הווקסלר, גם ממוריטן, וכמעט...
אצלם לא מתורגם ל... הם עיוורים.
מה? אז אני... גם... גיבים עכשיו לחוקים החדשים כן. בדיוק. סוף יכולות.
זה לא כזה טריוויאלי אצלנו. הם בעצם אותו גודל? רגע, מישהו ניסה להיכנס למרתיחן שמישהו צריך לראות? שלח? כן.
כך וכך לראות, כך וכך.
כן, אבל איך אפשר להיכנס אליהם? מה זה?
נכון.
תראה לי, אתה יודע? אין משפט. ואז הוא שלח אותי לאיזה NTBI, משהו, אתר כזה שיש לו, שעד עכשיו קראתי ממנו את כל המאמרים וזהו, באתר הזה. From here as in this? No, I did it from my house.
מסדרת.
So I have to connect to my...
לא צריך לחדש שם פעם מחדש, זה היופי.
התקציבים שלך,
מה שצריך...
החדרת שלך קיימים.
נכון.
University computer,
זה...
Oh, okay, so Wi-Fi
אפשר
is
לקחת את ה...
I mean, it's not fine. I want to read it from my house.
רמה... למה? זה עדיין עכשיו.
רגע, אז היית חושבים על הלגטריה במצב המודרני? זה
טוב, אז אם זה נכון שאנחנו רואים בעזרת המוח לא פחות מאשר בעזרת העיניים, מה? Oh yeah. Almost. Okay. So if it's true that we see with the brain not less than we see with the eyes, then some people, a smart guy from the US, Paul Bacharita, mainly suggested to use the touch system that said, okay, if most of the representation is in the brain and it's there, then maybe we can make a bypass. We can take an image by a camera and encode information using another sensory modality. For example, if I want to have the letter V here on the book, then I can stimulate the electrode here and here. Okay. And we developed in the lab a glove that works also compatible with the scanner that can scan the environment and give you the images based on touch. But several years ago, we thought it could be really cool to try and encode visual information using music and using the auditory modality. So just to give you the gist of it, we can, for example, think about giving the letter V to think about it like as a radar scan from left to right. Now we have only 45 minutes, so I'm going to skip some slides. Okay, I think I have some idea based on the first part, what I want to emphasize and what to go fast. Okay, so let's say we have the letter V. Then we can do it in the following way. We can have a scale going down and then scale going up, right? And we can represent it V. Now let's say this is the algorithm we choose. Then if I want to represent the letter W, what should I do? Down, up, down, up. If I want the letter M, up, down, up, down.
But how do you do that for a three-dimensional thing?
There is no three dimension in the world. It's an illusion. We don't have 3D in the visual system. Okay, so how do we do 3 dimension? The world is 3 dimension, right? Our eye, the retina, is 2 dimension, right? So how do we do 3D?
Different, 2 pictures.
Right, the differences between the pictures. Let's do a little later. You asked a good question, and I won't be able to show you anything that I want to, but that's more fun. Anyway, let's do it. So put your finger here and close your right and left eye and do it when it's closed. When is it jumping more? When it's closed. So actually what happened is that we have 2 images that are not the same but they not overlapping.
So you do this for the 2 years?
Yeah, exactly the two with the 2 years, yeah. Actually, you localize things in 3D by comparing the inputs from the 2 years. Exactly the same thing. So what you do is you compare the two images. You get two different images and you compare them. And if the overlap is big, you know it's close. And if the overlap is small, you know it's far away. So when you go to see Avatar and you want the frog to jump on you, what you do is that you have two images of the frog very similar to the two eyes. This is why you have these glasses. And now you start making them more and more far apart. And you get the illusion that they jump on you. Okay? So actually what the brain is doing, it actually proves this point very nicely. There is a reality of 3D. It falls on a 2D surface. And the brain creates an illusion of 3D that reflects the real 3D in the world. Okay? Now, if this was true, then how come when I close one eye, I can still see 3D? Exactly, because we have also monocular cues. Monocular cues are cues that we can use only in one eye. So, for example, it's like stand-up, you know? You sit in the first row, you are doomed to be bumbered. Let's look with you. You are closer to me, so you are hiding her, and you are hiding her, so I can do the depth. Also, you are brighter, and you are so on and so forth. In the visual sense. Maybe also in a different sense. I don't know. Anyway, so I can have also monocular cues, and monocular cues are available here, right? So actually we can do it. Also we have some, we played a little bit with having two inputs from two cameras and playing with it, or showing the difference between the two images.
You
and so
also
on.
have some methods of giving color.
Yes. Yes. Actually, I can already say this because I plan to do like a 10 minutes demo, and I'm not going to do it because I want to jump forward. You can think about the way we thought about colors is that we can, it was actually the first color SSD, and you can, for example, put the V to be played, or the W or the M to be played by different musical instruments. And teach them that always this musical instrument is this color, and a different musical instrument is a different color. So they can separate between the colors.
Like musical instruments also give you tones, just different harmonies. That's how they're doing it.
Yeah, exactly. The timbre in the music is by the different. Let's not get into a music lesson, the physics of music, but basically it's different relation of the overtones. So you have the basic principal tone, and you have some overtones, and different musical instruments have different ratios as the different overtones. So yeah, so the bottom line is we can teach also colors. So part of this study, we asked what happened when a blind person that didn't see for 10 or 20 years learn now to really do the color on a visual image using this algorithm, or to ask a different question: what happened when someone that never had an eye developed? We have a couple of these subjects that have anostalgia, that their eyes never developed, so they didn't see even one photon of light in their life, and they are amazing preparats, okay, we are in a science lesson, to study the development and the selectivity of the system without any visual experience. What happened when you never have visual experience? And now, let's say we are teaching them to sort colors. So they know the concept because they are interacting with sighted people, but now they can do it, okay? One philosophical and also in terms of consciousness is, what do they experience? So we have now a new project in the lab deal with this, and does it dynamic in time? And actually we found that people that are late blind, after extensive use, start to hallucinate the reality. So they would close the door and the door would jump on them. Okay? But what happens to people that never experienced vision? And so on and so forth. This will come soon. Okay, so let's, I suggest we move on. Okay, Elizabeth? Okay, so let's keep all this, just to say that these things already happened, it happens also in nature. So for example, echolocation and so on and so forth. I want to very quickly share with you two sensory substitution and the imaging work we did with them. The first is a minimalistic sensory substitution. So the idea is that we can develop a system that is holistic, that tries to represent vision and the topography and the colors, and this we did in the iMusic project, and we are still developing it to include more and more aspects. The second would be to say, now the problem with this approach is that it's more difficult to learn. Okay, so if you want to analyze, and I will show you a movie of this to look at you and see that you are smiling. You're not smiling now. And you're smiling, then I can also do it, but it's much more difficult because I have a lot of noise and I need to separate it from the background, etc. So the other types of sensory substitution are aimed for mainly one task. For example, the icon was developed only for navigation and mobility. And now the question is, how can we do it? It's very simple. And then what type of imaging work can we do with it? So this is the device, the actual device. It can come on the cane and give you a prediction for 5 meters or projection to upper body while the cane is on the floor. It can be a standalone. It's up to 5 meters. It's used infrared, and it gives you the input either in vibration or in sound. Okay? And actually, it can give you both. And the nice thing is that if you do it with sound, if you do it with vibration, it's very nice because you don't need to. You can leave your ears, open to the environment. The nice thing if you do it by audition is that you can run all these experiments in the scanner. So you can record, for example, what happened when somebody navigated a given maze, or you can give him a new maze that he navigates with exactly the same principles of higher vibration as things, or higher sound as things get closer, and see what happened in the brain when he's navigating using this input. Okay, so let's keep all this. And I just want to show you a congenitally blind that go into a maze, and after 2 minutes of experience with this is Shlomo. He was born blind. The speed is 2 times the reality, but it's still very quick. And you see, he found his way out just by the vibrations. Okay. And this is true. We had here a group of sighted doing it with blindfolds, of blade blind, of congenitally blind, and so on and so forth. Okay? Now, we also have all this very complex environment and also virtual environment. So I can show you one experiment that was done by Shachar Mandelbaum in the lab. You can see here a maze that you navigate through only with the 3 arrows, left, right, and straight, and you get this input, this very basic auditory input. And you navigate, and later they had to repeat and draw the environment and avoid obstacles and so on and so forth. So now you can do navigation with vision. You can do this experiment just by vision without the sound and try to navigate. You can do it by audition, and you can have it with people that have visual experience but now doing it with a different sensory modality. We're doing it with people that never had visual experience. And I want to show you only 2 results from this project and then move to the other project, because there we have like now already 6-7 years of a lot of imaging that was verified by several labs, and I can say it more convincingly what I want to share with you. But it seems that also here. So the 2 results I want to show you is one, a behavioral result, which we think is really cool. Using this virtual environment, we try to do a simulation that tries to understand what is the optimal distance for mobility to give them. Because if you give them, for example, 20 meters, then maybe you give them a lot of irrelevant information for mobility. And if you just give them 1 meter, like the cane, maybe you are limiting them because, for example, if they come to a new environment, they cannot explore it, or they can explore it in a very discrete way. So what we did in this experiment is that we actually tested what's happening when they come to a new environment where they have no device, when they have a device in the distance of the white cane, when they have vision, or when they have 5 meter distance. Or it works actually also with 4 and 3 meters. It depends also on the environment, but this is the numbers. And what is really cool is that when you are with no device or with a short distance device, what you will do, you will go left, right, or straight or right in different trials until you hit a wall. If you're unlucky, like in this case, you would choose left, and then you stick to the wall until you find the door and go out. Okay, or in this case, he was a bit more lucky, he went straight, and then hit this wall, and then went right. If he was doing left, it will be very long, okay? Now, what do you do in vision? For example, let's say this door was open, and I'm coming to this class for the first time. I would stand here and scan with my eyes. Vision is a long-range distance sense. And this is the beauty of vision. So I would scan, I would see this door open and go straight. And with this 4 to 5 meter cane, they would do very similar mobility in a lot of environments like vision. So here we have like a behavioral result of what would be a good way to substitute vision with audition. The second example, the second thing is that now we can go and put, and this is really like fresh from the oven data, is you can see that now we can characterize the navigation system from the visual modality. So we can do this experiment by vision and couple it with retinotopic mapping, and now go to areas that are mid or foveal and some other areas that are part of the dorsal stream for navigation. This is used for navigation. And now we can look what happened, and you can see that in some areas, that some areas can very quickly be recruited, and after some training, they are even being increased by responding to the auditory modality selectively to the mazes that were learned. So actually, we got areas that are almost reaching half of the visual responses to navigation, but now that it's based on sound information, and they have only several hours to train on them. And this was also happening in people that are sighted, and they are using blindfolds, just like I showed you here. Let's say I would now close your eyes and let you do this experiment and scan your brain while you're navigating. The area that will be most recruited for this, selectively for navigation, so we control for the auditory input and do it in a different context, it will be exactly the dorsal stream retinotopic area that are used for navigation. Okay? So this, first of all, show us that the visual system, is actually can be driven in a matter of hours. I didn't show you, but when we started the experiment, before they knew the algorithm, we gave them these sounds, and there was zero activity there. There was no activity at all. Only when they learned to use these sounds for navigation and to go out of mazes, and to navigate in some scenarios, we had some other, like the Morris Water maze and some other environment, only then the visual input arrived to the visual system and trigger it in a selective way. And the second type of result is that even people that were born without eyes and never experienced vision and didn't have this system to develop to vision, also recruited to the same way. My prediction is yes. In the blind, yes, but it's not an easy experiment to do in scanners. This is the beauty of the virtual environment.
But then also people who never experienced vision had some experience with exploring the environment with their hands and sticks. So
Definitely, the question is whether the body system should develop now this selectivity, or it will be the same neuronal machinery that we call vision, but I call it a machinery to perform given tasks that usually are being carried out by vision. That's maybe the difference. And what I try to argue is that several hours of, if you manage to teach to do the task that is in the basis of each network or brain selectivity or map or topography, then in a question of hours or tens of hours for different tasks and functions, you can recruit them to process as strongly. When we see like an area lighting up with like 50 voxels are firing, it means that hundreds of millions of neurons are working like crazy now for audition in this V1, V2, V3. in the dorsal stream. Okay, so this is now, but this is unpublished data. Okay, so I want to show you some stuff that we publish, and there is also replication from different labs, and this is more with this iMusic algorithm and several other algorithms. And basically, subjects can perform quite complex tasks using this, both in virtual environments and in real environments. I will actually just show you 2-3 examples, okay? And I usually try to teach you to read something when I'm not showing it to you. And actually, we show that you can teach in about 10 hours. We developed based on the iMusic, but using a different symbols. I think I won't have time to show you this project, but it's really cool. It's like a musical morph that is optimal in time and in terms of the ability to distinguish between characters. They managed to read like a Twitter in less than a minute, only based on this musical input. Okay, so I will just try to show you that it's also with the regular characters. In a matter of 2 hours, you can read any 3-letter word. Sighted, blind, anyone. Okay, and then we'll do some imaging with this. So just to give you an example, the line going down, and you can see the pentatonic scale going down. This is a line, and this is a line going down. It's very pure. Okay, a vertical line would be something that gives you a lot of sound in a very brief time. So very dirty. It's like pressing all the keyboards of the piano at the same time, but for a very brief time. So actually now you can have all the angles, right? Right, and this is what is represented in V1. So very quickly you can do this orientation selectivity mapping.
Do this inside a scanner, there's activity in V1?
That's experiment we're right now doing. But it's a very good question. Okay, so for example here, what do you hear? Diagonal going up. Very good. It's very quick. Let me also. I thought maybe to skip it, but let's do it for 1 minute. We'll do it very brief. Okay, most of the chances you won't be able to do it, but oops, let's still try. Okay, I'm trying to prepare you for a first word. Can you listen that the horizontal line is more pure than the vertical line? Can you listen that it's going out of the dirt? Yeah? And now you're ready for your first word. Okay? T. Okay? Now I don't have time, usually I would train you a bit more, but I would still give you an exam, and let's see, maybe some of you can do it. If you manage to read the word without seeing it, write it down on the piece of paper, okay? Don't say it aloud so you can let the others. I will give you a little clue. The first and the second letters are the H and I, and then you have 2 other letters, okay? So try H, I. So I would let 5. Okay. How many letters we have? 4. Can you say something about third and the fourth letter? They're the same, right? Okay, listen to it now again. This is the word. How many of you managed to?
I guess it's not because I heard it.
Yeah, but okay. So, but it could be also something else. Okay, but it show you even if you didn't manage, and some of you I already see that managed to do it, you can get a sense of the lower part of the L. This sounds brilliant to you. And also what is nice is that you can keep the topography, so you can tell them that the H is down or high in the visual field. Exit sign. So also the notion of where things in the world can be very quickly understood. Of course, when we now have a real stimulus like the image of Marilyn Monroe that you can do, but it will take you much more time, okay?
And everybody who's tried has managed to learn.
Everyone. That's amazing. We had like now hundreds of people that go through the lab for behavior, for imaging, for the sighted, low vision, late blind, congenitally blind. We never had one person that didn't manage to read after 1 or 2 hours any 3-letter word. And now, I just want to say one thing about it. When there is alternative way to restore vision would be to put you chip in the brain, in the eye. So it's working for about, in principle, for the 100,000 people that lost their vision because the generation of the photoreceptors, okay? Out of 40 million. You can implant so far about 160 to 100 pixels. Yes, it's glasses. Then there is a chip that stimulates the ganglion cells directly. Okay. Now this surgery cost, and we worked with some of these patients, about 100,000 euros right now. And after 1 year, about half of the patients cannot read one letter. Okay. So we can talk about the reasons. Some of the reasons are computational, it relates the code, it relates some other stuff. But I just want to give you this as a reference to what it means that you can read any 3-letter word in less than 1 hour or 2 hours of training. It means that it really can give you some knowledge about the environment, or you can navigate a complex environment just based on a different cue. Okay, we can do also.
Stuff like that. Different fonts and stuff like that don't interrupt the ability?
Well, let's say we have a different font. We actually had experiments with this, and you can generalize to different fonts and different locations also quite quickly. You will need to do a little training, but yeah. Because again, if you will use a different L and it will be written a little bit differently, still the topography will be very similar, right? So if you learn the topography and you see a couple of examples of different fonts, you will be able to generalize. That's a very good question. Okay, let me skip all these other examples that you can very easily do and tell you that using these, people can learn to read, to recognize objects, body images, faces, facial expression, and so on and so forth. Now, if I have time in the end, maybe I show you one movie about faces and facial expression. But now I want to go back to this question of what shapes the selectivities in our brain. And let's start with the most basic division of labor that, it's a textbook division. We have here the dorsal stream and the ventral stream that you learned in the previous lessons. Division, you have in blue this auditory system. Here we have the homunculus for touch, and here for motor. And there is a division of labor between the senses, right? Right. And we must have visual input in order to develop selectivities in this cortex, right? So this is the classic model, the brain as a sensory machine. Now, what happened to this visual cortex when I don't get visual experience? Or what happened when I had visual experience, but now I'm doing it with a different sense? Okay? So let's see some data. Okay, I will skip this. I think you learned it already. What is the concept of brain plasticity? And we have this classic experiment that's showing that this is irreversible, can be irreversible. The dominance to the right eye or to the left eye in primary visual cortex. That's the class, right? You learned this before. No? What? Okay, I have to learn to teach you this if not. This is the experiment that was part of the Nobel Prize for Critical Periods by Hubel and Wiesel. Okay? What is the experiment? Let's say you were supposed to know it by now. So let's do it. What is cortical plasticity? Changes that occur in the function and organization of the cortex as a consequence of experience. This is one definition of plasticity. Experience can be anything. Learning to operate in a world without vision, learning to see using the ear, learning to read, learning whatever. Okay? Learning a new language. Now, the most prevalent concept in cortical plasticity is the concept of critical period. Where did it was born? First of all, it was born from animal studies, and we need to see how much You know, there is an assumption in neuroscience that everything that happens in animals also happens in humans. I would argue this is not true, okay? But it's also not unrelated to each other. So this is our luck. But some cases, it might be that cortical plasticity or other plasticity or critical period, the windows would be different. And we have functions, for example, we all know that we can learn a new language in the age of 50. It's more difficult than in the age of 4, but we can still do it. Some people do it, okay? So maybe we need to think about cortical plasticity as tied up to a given task. Maybe it's different between animal and humans. And maybe it's more difficult to change, but things that are bound to critical or sensitive periods, but with some big effort, maybe you can still do it. Okay? Great. So what was the experiment that was showing this is so rigid and so limited, the critical period? The experiment that was done by Dewey and Wiesel was following. Let's say I'm now putting a radioactive label in one eye, but not in the other. Then I let it go into the system, and I sacrifice the animal. And I look now at the selectivity to the eye that was labeled or not labeled, and I can see that there is ocular dominance columns in the brain. Okay? So it means that this column responds to the radioactive label, and this not, and this yes, and this not. We have stripes, we have columns that respond, and we know now that it's true, that V1 responds more to one eye than the other eye. And then the next column would do the opposite. And now if you do this experiment and you deprive one eye, you would see that the other eye would go over the real estate of the deprived eye. And now you remove the blindfold, and this is not reversible. The other eye will be blind because there was competition. Gnurus and Z1 were recruited. So this means, and if you do it in the age of 1 year, nothing happens. Okay? You label, there is no changes in the cortical columns. And so first of all, it might be, so this is the result. It was replicated many times, and it means that there is some notion of critical periods. But does this mean that all the functions have several weeks of periods? Does it mean that animal and human behave in the same way? There was no manipulation here that tried to force the animal to use the eye that was blind. If I'm forcing it and patch the other eye and do some rehab and training to the other eye, will I still be able to reverse the picture or not? There is a lot of open questions. This is what I want to raise here. Okay, we take it. Okay, it's an amazing work. And I even met Wiesel, and he actually gave me my postdoc fellowship, and he's an amazing person. But there is still a lot of open question here that I want to try to raise, and I want to give you some examples. So first of all, let's go into the case that a blind person gets this word or get a face, and they need to recognize the facial expression, and we put them in the scanner. What do we see? And I'm here only talking about people that never experienced a photon of light. Zero visual experience. So actually what we did was to take the methods, the image that you saw here is based on averaging of a group of such blind individuals. And we took a method that was developed by Uri Hertz and Noah Charia, both of them ICC students, former, both graduated. And we published it in PNAS and J Neuroscience on the work that was dealing with topography in the motor system, some other sensory system, and the auditory system. And there we found multiple maps and mirror symmetry principles that was shown in the visual system to be also in all of these other systems. And the result was the method was very simple. You, just like in retinotopy, you organize, let's say, the body parts one next to each other, and you do and so you have a very organized order based on time, right? And then we do a Fourier analysis, and we retrieve the phases, and the phases give you the body map, and you do it from different directions, and you do some statistical correction, but then let's all do the deal. But now, let's say now we have a situation in which a blind gets this information and try to build the word hill, or to build the facial expression, or to build an object. Okay? It takes them about 4 or 5 seconds. I don't know if you notice. It takes a little bit more time than vision. Okay? That's another advantage of vision. It's very immediate, right? So it's like vision in slow motion. And now let's look at the phases, repeat each stimuli many times with some baseline between them. And now let's see what is the information processing in the brain when they start perceiving the input and when they manage to build the right word or the right facial expression or the right object. And what we see, I think, is really, really cool. We see this picture. So this is by the way, it's a movie of the phases when we stimulate, when subjects move the feet or the hands or the fingers, or each of the 10 fingers, 7 facial expression. And you see here a beautiful map in the basal ganglia that goes just like pencil homunculus that is in S1 or M1 from the feet into the legs. And actually, based on this, we can, in a very high level of accuracy, in a different experiment, predict what is the movement based on the activity in this map. Okay? But now let's go back to this experiment of seeing with your ears. Okay? That's the maximum voice. Okay, never mind. So what you see here is that first of all what happens, and this was true in all the stimuli we use. Actually what is cool is that it's true also in sighted. But it's even more amazing when it was done without visual experience. In the first second, the first CR, the brain goes to A1 and do a very quick probability analysis of the frequencies and the stress and the volume. But now when you start to build the topography to understand that this is an H, this is an I, this is an L, the information travels, and the time that is correlated with the recognition is when the visual system is active. So it's the same system that builds the topography in vision, builds the topography also when you never had visual experience. This is already says something in our view of the organization of the brain have to be modified, okay? But it's even more than this, because we know that beyond the retinotopic areas, and this is most of the work we did and replicated, and now there is a lot of like 10 labs that replicated all these results. We know that beyond the topography map, we have areas that are specialized. For example, let's start with the first area. This is an area that analyzes people, other people. We have, by the way, specialization for navigation. It's called physical place area, but basically it's analyzing the environment and helping us to get cues for navigation. We have for faces, it's a very important social cue for us. For reading, which raises some very interesting questions. By the way, if it's critical period and we start learning to read when we are 6, how can we have an area that is very, actually from all these areas, the area that is most consistent across different individuals, even in different languages and different continents, is this area. How can it be? This is not work. It cannot work in evolutionary mechanisms. It has to be something else.
So when people learn to read after the age of 6?
Also when people learn to read after the age of 6, and also when people learn to read Braille in the age of 6 when they are blind, or like we showed in this week publication, when sighted learn to read using Braille. And we actually also interact with EMS. I might show you some data with this and show that this is a causal relationship, not just correlative. Okay?
It's in no way something related to semantics that is not reading?
No, because we can control in some other labs that there is hundreds of papers over this exact question. So if you just listen to the words, you don't get any activation. Or if you do, in one experiment, for example, we gave the heel, or like a new word, then we led them by semantic hear 2 words, and then they have to choose if the word they heard using the ear or both they heard with the ear. One with a sensory intuition and one with semantics. And we saw a reduction to even below baseline with the semantics. So it's only when they build the topography of the symbol and connect it to phonology. So actually using this type of experiment, we can actually understand what really a brain area is doing by all this manipulation. When we understand what is the task, we can actually understand what is the computation it is doing. And what we think now is that this area is responsible to take any geometry, any symbol geometry, and assign phonology value to it. and there is actually we found homolog area in the other hemisphere that do exactly the same but for quantity. So symbols, to take a symbol and connect it to a given quantity. And this goes with a different type of connectivity to a different brain area.
In place of something that is not reading that should activate this area, sometimes it's a task, just like you need to...
Exactly. So now you have a good hypothesis to say, okay, now when I listen to words, shouldn't activate. When I now teach arbitrary symbol, and now, why don't I show you the sum of the data, okay? But let's start and then let's continue. This is the next area. Navigation, faces, reading, and then all the tools and the objects that we use. And there is one more area that is very important. It was actually the last to be discovered from this slide. And this is an area that analyzes bodies of people. Now, this area is very important. Why? It's very important because this is the main input. And if you look at the research, it's the only input into the mirror system. Okay? What is the mirror system? It's a system that was found by Rizzolatti. And basically, it's an area that has neurons. And here, it's an example with a very beautiful translation from animal study that was found also in human. Okay? But again, it doesn't mean that the system works exactly the same. Okay? But the principle looks quite similar in the sense that these areas have neurons that respond, or areas, or like populations of neurons, that respond in the same way when I'm looking at someone doing a motor task and when I'm doing it myself. And this, you understand, this is very important because, for example, when a baby learns to do something, when you open for the first time the phone, your iPhone, and press the home button, so they look at you and they do the same. And this is how they learn. And some people say this also is a very important system even for social cues. So for example, if I'm seeing someone that is smiling and I'm smiling and I'm feeling great, or I'm crying and I'm feeling bad and I see someone else crying, then I can connect my feeling to the feelings of others, right? And this system, as I said, it's very preserved in evolution. You can see. So the main input, how do we analyze other people's actions? By vision, right? We look to them. And this is very preserved in evolution. Let me give you a little clip. Yeah, and humans also can do it, you know. Of the other, okay? Not all animals can do it, by the way. There are some animals that cannot do it. But now, in one experiment, what we did was to teach blind to interpret, to learn other people's body postures and body images using the sound. And you can see here the first time that Hamutal is a congenitally blind, that we ask her to imitate a given
עכשיו למשל אני מבינה שזה אמור להיות קטגוריה של גוף מלא, ואני שומעת קווים אופקים, אז אני מניחה שהוא עומד ככה.
And now we replicated in this experiment exactly in the scanner, when we give there in one category a lot of visual body postures that she needs to judge. We give her the object, we give her to read, we give her faces, and so on and so forth. And now we ask where in the brain there is the peak of activity when we image the brain of all these individuals. And what we found out is pretty cool. This is if you do the experiment by vision and you just contrast it with rest. You just give the visual images and you don't do any controls. The controls I will show you in a second. And what you see is very cool is that the peak of the activity is in this extra-striate visual area with some preference to the right hemisphere. And you see that there is nothing in A1 because this is a visual experiment. Okay? Now we do the same experiment with congenitally blind that never had any photon of light experience, no visual experience. And you can see that there is activity in A1 and some areas around it. But the peak activity for these body postures when they do this task in the scanner is in the right extra-striate body area. And now when we look at the overlap, we see that the overlap bilaterally is in the EBA with preference to the right hemisphere. So even the visual extra striate area, the main input to the mirror system, is driven by a different sensory modality if you learn to do this task. Now what is really cool in this experiment is that we did also a single subject level analysis in which we had the sighted performed by vision, we had blind doing it by the seeing with the ears, okay? And now we look at the peak of activation versus any other category, versus faces, versus reading, versus all of this. And what we see now is that actually when we remove the group tag, we cannot separate the peaks in the brain from a congenitally blind or a sighted, okay? Okay. Now I won't have time to show you too much, but I will show you three more very quick examples. This is an experiment that Leora in the lab did, which she did exactly the same experiment, but this time with Braille versus other categories. Okay? And she showed that Braille individuals are actually reading using their visual word-from area. And this is true, now we show all the spectrum. It's true if I'm going to take, like in this study, congenitally blind that learn to read Braille. But now we showed from this month that together with our collaborators in Poland, several amazing Braille teachers that took and managed to teach sighted, which is more difficult than blind actually, to teach them to read. They managed to read very nicely with Braille, and we showed that activation is exactly the same place.
But why is it visual? If I'm listening to an audiobook, it's also...
No, because there you don't do, then it's only semantic. What is reading? Reading is to know that this symbol means this phoneme a, this symbol is da, and this symbol is etc. Or this symbol is e, and this r, and this is th. When you do an audiobook, you just listen to the semantic. You don't do this conversion from symbol to phoneme. This is reading. Actually, this is helping us to understand what this brain area is doing. And actually, when we look at connectivity, we can see that this area most resonant, have most resting state connectivity into phonology and language area. And when we look to the homology area, which has preference for digits and quantity, this area is connected even in congenitally blind, so there is some programming for the connectivity that actually triggers this task selectivity. This is a line of work I won't have time to share with you, but I planned originally. But okay, so let's see another example. This is actually the study. What was actually nice in this study is that the physical, you can see that in some cases, the symbol for letters, the phonology mapping, and for the number mapping, it depends on the context. So here, for example, this symbol is O, and in this context, this symbol represents the quantity 0, right? Or tenth. So now we created an experiment in which we gave the letters X, I, and V, using vision or using sensory substitution, but the context was different. In one context, the task was to assign the quantity and to look for the quantity 5, and one time it was to look for the phonology and assign the letter values. But the physical stimulus was exactly the same, okay? And what we showed is that, first of all, you see the peak of the brain when they treat it as letters in sighted and congenitally blind, and this is for numbers, and this is in congenitally blind, in sighted. So they actually overlap. And now we also had in this experiment a new method we developed in a different study, which is called partial correlation. Basically, it looks which connectivity one area has after you partial out the connectivity that a different area is having at baseline. So which area, when they elevate, they have a tendency to elevate with other brain areas, and when they go down, they have a tendency to go down with other brain areas. But we regress out the connectivity of a different area. And when we do this, we show that the visual word form is connected to the phonology and the language system. And the number form area is connected into the quantity areas in the parietal cortex. This month we talked about quantity and numerosity early on. So it means that we are born with a set of connectivity, but we can switch the senses between them in a question of hours. Okay? Maybe the most convincing, or the study that we pushed, I think the speed that this can happen is this study in which we, as I told you, we created based on the eye music like a different musical pattern that is have different shape, but it's driven from the music for different letters. And we were teaching half of the letters, and half of the letters we didn't teach. Okay, I won't get into all the behavioral results. Actually, what is really cool is we showed when we added a color, a timbre quantity into the letters, the speed of learning and the accuracy was much higher when it was the different letters were only differentiated by the shapes. Okay, but never mind about this.
But it was the same color all the time to that letter?
Exactly.
Okay.
So it's like we created synesthesia.
Yeah.
Okay, in a way. But the thing is, it was another cue, and suddenly you are doing it much better. But what is really cool is the following study. What we did was to take subjects that knew how to read Braille, and we let them listen to the letters. Now, half of the letters we took them out of the scanner, and for 2 hours we teach them only these letters. So what did we teach them? We teach them the mapping between the symbol and the phonology, right? And the other half, we didn't teach them the mapping. It was still noises that they knew how to build the shape, but they didn't say anything to them. And after 2 hours, you get the activity level in the reading center to be exactly the same as the Braille. And the letters that were not taught were left in baseline. Okay? So I think this should suggest that really it's visual experience. Maybe the last movie I'm going to show you is in the tool area. We actually did a lot of area of research here in a recent study by Zohar Tafanda. For example, we showed that at rest, this area, this tool area, has a unique connectivity only to the body input from the body, from the arm and the hands. So it means that this area that is tool selective, visually, already have inherent input inside it. Yeah, this is not blind. That is driven, this area, by input from the body. It shows also, again, how much all the senses work actually together, like the example of the McGurk or understanding phonology. And what we showed here, that first of all, you can see here the spectrogram of the sound of a horse. Yeah? So you see the topography in the sound, and you see the activation again. There is some differences, but the peak of activation is exactly the same area. But what I want to show you and to end is a really cool experiment that we did using TMS. This is quite rare because most of these specialization are ventral, so they are not accessible by TMS because they are in the lower floor of the ventral stream. Okay, but actually the tool area is very lateral. So the most dorsal part is laterally, so you can access it using TMS. TMS, did you hear about TMS? Did you see? Okay, so I just show you, for example, TMS over Broca area. There is two types of TMS. One is online, which you stimulate something and you interrupt the function while you are doing it.
Virtue, ocean, ambulance, line, It'd be hard to say the words.
Just couldn't get it out.
Couldn't get it out.
So this is a live demonstration of Broca area. A different way to do it, if you want to do a more slower task, and actually it's more safe, is to stimulate not in high frequency but in low frequency, less than 1 hertz for 10 minutes, and then you basically deactivate for mechanisms that are not 100% understood. It's probably not exhausting the vesicles.
Yeah, but don't you shut off a lot of stuff around it?
Sure. So it's not that clean, but actually, if you move like 1 centimeter forward or backward, you lose the effect. So it's very specific effect. Actually, it's rare. It's actually, people get the impression that TMS is like a piece of cake tool. Usually what will happen is that you do the TMS and there is zero effect on behavior. So when you have effect like this, you can study the area really. But what I want to show you is this other method of doing 10 minutes and then you have deactivation for 10 minutes of the function in this area without doing TMS in these 10 minutes. And this is you see here a late line individual. See the glasses? She's actually one of the subjects.
Object is detected.
She detects the object.
Tape.
She recognizes the tape, even though the resolution was really bad.
No connector?
No connector. Was really bad.
The
Now
object
she
is
has 10
now in
minutes
front
of
of you.
TMS into this
Now you try to
based
find
on the
that
MRI
edge.
that we do here.
Okay, there's the edge.
And now she can go on for another 5 minutes. Never she cannot even localize the region of it. So it shows like a causal effect. We also show that the separation to ventral function and dorsal function are maintained and so on and so forth. So just to summarize, the visual areas, including the reading center, the body center, object and tool, movement, dorsal stream, faces, navigation, appears to be related to the task regardless of the sensory inputs. So the selectivity is to the task, not to the sense, not to vision. It can be independent with enough training to visual experience. So people that never had experience in reading in this modality and recognizing objects, in doing the mirror system stuff, can still recruit it. You can learn this age in different ages and still get this effect, and so on and so forth. So this suggests that the brain is not a sensory machine, but sensory independent task machine. There is some studies from animal I won't have time to show you, but you can do this experiment and show that in deaf cats, for example, there is vision can drive the same area according to the task, and you can, in these cases, deactivate this area by cooling and show the effect to be causal too. So it's the same type. And I would just, in terms of rehab, I just want to share with you a project that we just started, and it's actually an ongoing project now. Now, based on all this input, what we try to do is to, it's a 5-year ERC project, to build a multisensory, it's only part of the project, the other part I actually showed you the results of it, to build a multisensory bionic eye. So the idea is that you will have a glove, you will have some auditory input that can give you localization, can give you color vision, and so on and so forth. And the idea is that you will get all this information in the same time. So before the operation, you will train to interpret images that come from camera, the same camera that you will get in the implant. Then you will train on all this. Now you will get the implant, and this will help you to understand the mess that the electrodes are creating in your retina that's supposed to simulate vision. And at the same time, you will get all this other input. And then we'll see if the rehab is faster and whether indeed we recover all the awakening of the visual system using this approach. So this is the rehab part of the project. Okay, I think I will stop. Oh, we're out of time. So if you have a stressing question, you can come and ask me. And yeah, you have one?
You can also develop like health experiments with additional sensors, like infrared, I don't know.
Wonderful, wonderful. So actually, it's a great because usually my first slide of talks. Let's see if I have it here. If I have it, I will show it to you. It's 1 minute. It's 1 minute slide, and I think this is another really, really exciting direction that. I'm going to show you what was done already. Okay, it's really, really cool. Let's see if I have it here. I don't. So I will just draw it to you in a. So Nikos Logothetis, Nicolelis lab in the US. What they do is the following. Actually, we have one project in the lab that we try to give a nuisance to sighted people, but this is very preliminary, so I can't share with you. But there is a paper already published, actually 2 papers, that I think are amazing. I love them. And so check them out in YouTube. The idea is the following. You take a rat, you go to the homunculus, and you put an electrode there in the somatosensory system. An RF electrode, just like the bionic eye. Okay? But now, instead of giving the input from a camera for vision, okay, the rat is the size of the right? You couple the stimulation to a camera that is an infrared camera. Just like the IK, by the way, that can sense difference in thermal or infrared input that is not in the sensory scale of this animal. And they do a little bit of training, And now they close all the lights, and they put several food sources, and only in one of them there is a infrared input that's marked. So there is a lot of location, but only in one of them there is food, and it's marked by the infrared. And now the rat learns to interpret the electric signals into his body system to go to the right location of this new sense modality. So we are in an area, and you can do it, just like we showed it, all these things that you can do, basically restore a given function invasively, you can also do non-invasively. And there is a very interesting, I can describe to you, like for example, from our experience, 2 questions I would love to test in the next couple of years that we still didn't do with this new senses experience. So for example, let's say now you start to do this, you have this camera and it gives you the sound. Or you have this glove and it gives you input on the body of the feet. What happened in the first hour or 2 is that you experience the input coming from your body. And I think this is what happened in this rat. In the beginning, it feels like somebody was stimulating his hand or his finger. But as you learn to use the cane or the SSD, you learn that it represents things outside. And what happened after several hours